Hello, my name is Alexandra de la Cruz, and this is my PowerPoint on cytoskeleton movement. The goal of this presentation is to learn about roles and the importance of cytoskeletal motor proteins. So to start off with a disease caused by a kinesin mutation, I want to go ahead and talk about hereditary spastic paraplegia, also known as HSP. So to go in depth about the specific mutation, it is when two catalytic domain mutations, specifically the R28OS and the K253N, reduce the microtubule binding affinity of the kinesin head domains. So that is the specific mutation that takes place in order to cause HSP. In addition to the kinesin MT binding and the coil-to-coil depolarization, HSP mutations affect the motor stepping transitions and processively lead to this disease. It's important to note that specific mutations in the kinesin 1 family motor, also known as the KIF5A, have been reported to occur in about little to less than 10% of patients identified with HSP. HSP is a heterogeneous illness that causes um, overall and progressive damage or accidents into the corticospinal tract. So here in this image, I wanted to go ahead and talk about the structure. So in letter A, it demonstrates the MT bound single head domain of kinesin. That's where you can see that blue, and over here in the red, you can see where it's identifying the head. And in letter B, it demonstrates the head domain residue, also known as the 36-1 ALA, that is located at the coil stalk region, right here in letter B. So my conclusions for letter A is that it demonstrates that over the time the length of the polymer is increasing. However, it's only when the buffer is being used. So you won't be able to see the changes when the buffer is not being used. Furthermore, the effect on the microtubule length as a function of time when the tubulin and buffer are replaced. They are first identified by an identical tubulin, and the buffer solution is being demonstrated as well, as you can see there in letter A. Moving on to B, it demonstrates that the polymer is increasing only when you add the kinesin 1. Therefore, it's evident that kinesin 1 is overall favoring depolymerization, showing and proving that kinesin 1 is facilitating the depolymerization. Furthermore, image B also demonstrates when the tubulin and buffer solution in the chamber are replaced with a solution containing tubulin and the M-type, the kinesin motor protein is shown to be kin1, as you can see there. So kin1 is very, very much needed for depolymerization. So right here in experiment 2, or letter C, we can see that the T-stabilized microtubules are incubated in 4 distinct chambers. As you can see, they are labeled respectively. You have your ATP, 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 and then lastly your AMPP and P. And those are 4 distinct chambers. So here you can see that the supernatant and pellet fractions are separated by SDS, and the gel was stained to reveal the position of the tubulin. Therefore, we can observe that the kinase heavy chain tubulin is still in its pellet form. So therefore, when kin1 is added, polymerization into the tubulin takes place. When kin1 and the AMPPNP is added, no depolymerization is happening. This is because the tubulin is still in its pellet form. This shows how important and significant ATP for kin1 activity is, since kin1 requires and depends on that ATP hydrolysis in order to facilitate the polymerization. And once again, this shows the same thing in letter B over here, just basically stating that kin1 is highly depending on the ATP hydrolysis. And these are my references. Hope you enjoyed.